A first principles approach to muscle adaptation, the opposing concepts of hypertrophy growth and hypotrophy. Atrophy must be grounded in the molecular biology and cellular mechanics that govern muscle fiber size. This framework breaks down muscle change into three fundamental, non-negotiable principles: the protein balance, the anabolic trigger, and the catabolic signal. 1. The zero-sum principle. Net protein balance. The fundamental truth of muscle size is that it is governed by a simple, zero-sum equation within the muscle fiber myofiber. Muscle mass rate of muscle protein breakdown, MPB rate of muscle protein synthesis, MPS. Hypertrophy occurs when the rate of MPS consistently and significantly outweighs the rate of MPB, resulting in a net positive protein balance. Hypotrophy atrophy occurs when the rate of MPB consistently exceeds the rate of MPS, resulting in a net negative protein balance. Muscle size is, therefore, not a measure of effort but of the cumulative difference between these two opposing molecular processes over time. 2. The hypertrophy principle, anabolic signaling and cellular infrastructure. MLSEC. Hypertrophy is triggered by signals that demand the physical expansion of the myofiber's contractile machinery, myofibrils. This process involves a master regulatory pathway. The anabolic trigger TOR. The primary molecular switch for MPS is the TOR mammalian target of rapamycin pathway. Leading research, including studies frequently conducted at institutions like Harvard and Yale, identifies three primary inputs that activate this pathway to initiate growth: mechanical tension, the shear tension produced by lifting a heavy load. This is considered the most potent signal, disrupting the muscle cell's mechanosensors and forcing adaptation. Amino acid availability, specifically, the presence of sufficient leucine branch chain amino acid acts as a signal to the cell that adequate building materials are present. Growth factors, signals like insulin-like growth factor 1 IGF-1, b. The cellular infrastructure myonuclei sustained. Long-term hypertrophy requires the muscle fiber to increase its genetic control over the expanded volume. This is achieved through satellite cells, which are resident muscle stem cells. They are recruited to fuse with the existing muscle fiber, donating new myonuclei the cell's command center. This adherence to the myonuclear domain theory dictates that each nucleus can only support a specific volume of cytoplasm and protein. To grow larger, the muscle fiber must acquire more nuclei to manage the increased protein synthesis. 3. The hypotrophy principle. Catabolic signaling and degradation pathways. Muscle hypertrophy atrophy is the rapid result of removing the necessary anabolic stimulus disuse or introducing a potent catabolic signal. Illness, a absence of tension, the disuse signal. The removal of mechanical tension sends an immediate signal to the cell that the existing protein mass is unnecessary, thus eliminating the primary stimulus required to maintain the positive net protein balance. B. The degradation pathway. Ubiquitin proteasome. The body uses highly efficient, targeted systems to break down and recycle muscle protein when it is not needed or when the organism is under stress, for example, starvation, severe illness. The key pathway for targeted protein destruction is the ubiquitin proteasome system. Ubiquitin acts as a small molecular tag applied to muscle proteins that are slated for destruction. The proteasome then acts as the cell's recycling plant, recognizing the ubiquitin tag and rapidly degrading the tagged proteins actin and myosin into individual amino acids, which are then used for energy or other bodily functions. Research on age-related muscle loss or sarcopenia, a major focus at institutions studying geriatric health, reveals that this catabolic signaling pathway becomes chronically overactive with age, driving the inevitable decline in muscle mass.